So it's actually split up into two stages, one called transcription and one called translation. So let's really delve down now into the detail. Transcription is the first part. It's where you take your DNA and you copy that code onto a molecule called messenger RNA. Translation is then when that messenger RNA code is translated into the amino acids that we want using some molecules called transfer RNA molecules. So just mentioned two types of RNA there, messenger RNA and transfer RNA. What is RNA? That's probably a question we should answer first. Well, RNA is a very similar molecule to DNA actually. There's just a couple of differences. You can see them in the diagram here. RNA is single-stranded, when DNA in its normal form has these two strands, a double helix. And RNA, very importantly, uses a different base. Remember the four bases in DNA, A, T, C, and G? Well, in RNA, there is a change. You don't have T, you don't have thymine, instead you have uracil. So there's a different base in RNA. Now, proteins are assembled in the cytoplasm of a cell, but the genes are stored in the nucleus. So we've got a problem. How do we get that code from the nucleus into the cytoplasm and the ribosomes, which is where the proteins are made? Now I suppose the DNA could come out, but remember the DNA is this massive long supercoiled molecule, all in its genes, all joined together. How are we going to take one gene out and then put it back again in the same place? You can't do that. What you do is you basically make a photocopy of the page of the code that you're interested in. Imagine that. Imagine all the instructions for all the Lego models in the world had to be stored in a very special library. You could go there, you could photocopy the pages you need and take them home with you to build your model. It's a bit like that in the cell. So what we've got here is we've got all the instructions in the nucleus. We want to take a photocopy of the gene, and then that photocopy can go out into the cytoplasm and the code can be read and the protein can be assembled. Now that photocopy is what we call messenger RNA. So here is our DNA strand. It has been unwound because remember we only read one side, and we're now going to form our messenger RNA copy of the gene. Remember complementary base pairing? If there was an A on one side, you get a T on the other side. If there's C, you get the G. But remember, we're dealing with RNA now, so wherever there would be a T, you actually see a U, a uracil. So here's our DNA code, and this is the messenger RNA code that is formed from that DNA code. Then it is nice, small, simple molecule which can travel out of the nucleus into the cytoplasm for the next stage, which is called translation. So we're halfway through the process now. Now in translation, you need two things. You need a ribosome and you need something called transfer RNA. Now a ribosome, we talked briefly about it in the cell structure video, if you need to look back at that one, but it's a very small particle that exists in the cytoplasm, and its job is to take that code, read the code 3 at a time, read the codons that we talked about, and then to take another molecule called transfer RNA, and that's where the amino acid will come in order to start forming the protein. So transfer RNA is this kind of cloverleaf shaped molecule, and you can see at the bottom there are 3 little bases which are going to be complementary to the codon. We call them the anticodon. And at the other end of the molecule, you can see where the amino acid will bind, the corresponding amino acid will bind. So this makes more sense when you look at it as a diagram. And here's the ribosome, here's the messenger RNA, and the ribosome will move along the messenger RNA, reading 3 bases at a time, reading the codon. And at each codon, the corresponding transfer RNA with the anticodon that matches the codon will come in, and the amino acid will be attached to that transfer RNA. Then the next transfer RNA will come along. You get 2 coming in at a time. You can see the anticodon, UUU, is the opposite of AAA. Remember, we're dealing with messenger RNA and transfer RNA, so both these molecules are RNA. And then the first transfer RNA gets released and the ribosome moves along. Next transfer RNA comes in. RNA binds to the amino acid binds to the last amino acid. That transfer RNA disappears and the process continues. And it's like a little factory production line. Ribosome there, reads the code, GUC, transfer RNA with the corresponding anticodon, CAG comes in, the respective amino acid is attached to that. That then binds the rest of the amino acids, and we go along this little assembly line building our protein until you reach what we call a stop codon, and you end up with your amino acid sequence from the gene that was originally in the DNA. That amino acid chain goes off and gets folded up into one of those really cool complex 3D protein molecules. Then the messenger RNA gets released and can be reused, the ribosome can be reused, and the process can start all over again. So that is transcription and translation. DNA is in all living things. The triplet code works the same in all living organisms, and therefore we call it the universal code. This is the secret of life. This is how life works, via DNA, via protein synthesis. So it's such an important part of biology. When Watson and Crick discovered the structure of DNA, they discovered these bases, they went into the Griffin pub in Cambridge and they announced, we have discovered the secret of life, because they had. This is really the fundamental principles underlying how organisms work. And it's exciting that it's universal, because what that means is we can swap bits around. We could take a gene out of one organism, put it into another one, and that organism would manufacture the protein in the same way, even though it wasn't from its original DNA. So that's where genetic modification comes in, and genetic engineering, which comes up later in the course.